Hi, in this video tutorial I'm gonna show you how to search and retrieve the species-specific data on ligand binding and gene expression data as well as structural data. So let's start. Okay, so now we will use the ChEMBL database for searching the bioactivity data related to species-specific binding. And I have to say that ChEMBL is almost the one and only database that provides the species-specific binding data. So it's the first choice when you are looking for such kind of data. Okay, so after you Googled it, or if you know the direct address, you can just put it to the browser search field and open ChEMBL. So now I'm going to show you how to look for a species-specific ligand bioactivity data, and now I will use one of the common proteins. It's called retinoic acid X receptor gamma, and show you how to find the data on ligand affinity to human and mouse sequence variants of this receptor. So for the start, we just have to put the name of a protein here in the search field. So it's retinoic acid X receptor gamma, and write homo for Homo sapiens. And now we have the results. So as you remember, ChEMBL provides you results in a format of different columns. Here we have all results, which combines the different outputs. Then we have column for compounds, targets, assays, documents, cellular data and tissue data. Right now we need to select the targets. Okay, here we have 24 records for retinoic X receptor gamma, but for different organisms. Here we have the Homo sapiens, so its ChEMBL ID is 2004. So you can use right mouse button and open it in a new link. So as you see here, there are data on 239 bioactivity points, which can be the Ki values, IC50 values, and now also we will use the mouse or mus musculus bioactivity data for retinoic X receptor gamma. So you can open it. Okay, so now we have report cards for two targets, for human and mouse retinoic X receptor gamma. So first, let's work with the human version. So you see here the organism, Homo sapiens, and the preferred name, retinoic X receptor gamma. Let's go down, scroll down to activity charts. So here you see the bioactivity chart with different coloring representing different parameters that were measured experimentally. So most of the data comes from Ki, EC50 or activity measurements. Some data also is about dissociation constant, IC50 value, efficacy, inhibition, and so on. Now go to the second tab for mouse version of the receptor and scroll down to the same bioactivity chart. We will check the Ki values, since Ki values are independent of the assay type. So you should remember that EC50, IC50 values, they are always dependent on the assay type that were used to measure them. So they should be converted to Ki values before they can be directly compared. So open Ki, for mouse we have 29 bioactivity points, and for human we have much more, 116. Okay, so after a moment we have the whole table. So let's check the format of the data. Okay, so we can check the columns and which types of bioactivity data we have here. In the first column you have the ChEMBL ID, and you see the minimized formula. Then you have the compound key. Usually it's the name given by authors who measured this bioactivity and that were used in the paper. Like this original paper, this compound was called compound 36, or this one compound 12B. So it's an internal naming of compounds. Then we have description of kind of parameter that were determined. So in this case it's Ki, or inhibition constant. Then we have relation, it means that Ki equals to this standard value. So here we see the actual bioactivity. So this compound has Ki value of 20 nanomoles. At first we can sort the table according to standard Ki value. So let's put the lowest value which resembles to the most powerful inhibitor. So as you see, now we have the most powerful inhibitors at the top of the table. So this value is quite good. It means strong inhibition. Then we have converted Ki, so it's pKy. And this pKy value is calculated by using the negative logarithm from Ki. So the lower the Ki value, the bigger is the pKi value. So when you want to figure out which inhibitor is stronger, you have to compare either Ki value or pKi value. The lower the Ki value is, the stronger the inhibitor, but the higher is the pKi value, the more powerful the inhibitor will be. So they are oppositely related. Then we have some comments from original paper or from submission based. Then again we have the ChEMBL ID, but in this case for the assay that have been used to determine this bioactivity data. Then we have a brief description of the assay that was used, so you can find more data here. And also many other data such as organism that was used for the assay, ChEMBL ID for the target. In our case we have only one target, it's retinoid X receptor gamma from Homo sapiens, so ChEMBL ID will be the same here, as well as target organism and target name. And also we have here some data about the original publication, so we have ChEMBL ID for the reference publication. Okay, so now, as you see, we have a lot, 116 data points. We can screen the whole page, the whole results table, or we can open them at once here. So 100 is the maximum. So now you see much more. Okay, but more convenient will be to work with the standalone file. So we can download it here as the common separated values table. Put download CSV here, and now the ChEMBL will generate the download file. Okay, just now the CSV table with all the bioactivity data about the inhibitors of retinoid X receptor gamma has been downloaded. So now we can turn to the second page, which comprises the bioactivity data on inhibitors of mouse version of this enzyme. In this case, we have only 29 records, so we can easily visualize all of them on one page. And again, let's sort the table according to the standard value of Ki. Also, now at the top we have the best inhibitors with the lowest Ki value or highest pKi value. And then we have compounds that lack inhibiting power. Usually, it's widely accepted in the medicinal chemistry field that the cutoff value, which divides the group of actives or inhibitors from decoys or inactive compound, on the level of 30,000 nanomoles or 30 micromoles. So it means that if Ki value is lower than 30 micromoles, the compound is treated as active. If the Ki value is larger than 30 micromoles, the compound is treated as decoy. Of course, there is a kind of twilight zone from 10 to 30 micromoles. The compounds sometimes treated as actives or inactives, so if you want to be sure that you select only actives, use the cutoff of 10 micromoles. So everything below 10 micromoles is active, above 10 to 30 micromoles is a twilight zone, and more than 30 micromoles is inactive compound or decoy. Okay, so again we have a table with Ki values, and again let's download them. But in this case it will be the data for mouse version of the same retinoid X receptor gamma. So now it takes some time. Okay, great. Now both tables with bioactivity data have been downloaded, so we can use Microsoft Excel. It's the easiest option to compare the results. Okay, here I'm not going to tell you that it's the only and the most correct way of doing this, but it's an easy and convenient way of doing this. Okay, now we can open both files in Excel and compare them. Click on Open, and here select All Files, so your CSV files will be seen by Excel. So now you see them. Just now I have unarchivated these two files because they are downloaded as WinRAR archives. So now I can open the first one. So as you see, it's the file corresponding to mouse bioactivity data. And also, let's open the second file. doing the same. This time it will be for human, yeah, because it has much more. First of all, we have to recognize the data, because now everything is collated into one line. So select the first column, go to data, and change text to columns. Here, use a delimiter, and in our case, delimiter will be semicolon. So after you do this, you have different tables which resemble the format of original ChEMBL. Do the same for the second file. Again, go to data, text to columns, select delimiter and semicolon. Okay, great. So now let's sort them by name. Here, sort from A to Z, and of course expand the selection so all columns will be sorted as well. And now do the same for the second file. Again, sort them by alphabet. We can remove all the excessive data. So right now we need only bioactivity data, which lies here. So we can remove all other columns. We know that all the data are in nanomoles here. Okay, so now we have only standard value of Ki in nanomoles. And do the same for the second one. Remove everything beside the standard values. Okay. And now we can create a collated table, a spreadsheet here. Let it be the summary. So here we will copy the data from both spreadsheets. So here we have the mouse bioactivity data and past here the human bioactivity data. Okay, so as you can already see, we have bioactivity data for the same compounds. So for example, compound ChEMBL 1023 has the same Ki value both for human and mouse protein. But of course, it also has other values. What is the reason for this? Why we have many values? It's because there are many assays in which the authors measured the bioactivity of the ChEMBL 1023 compound. So it comes from different kinds of assays. So to directly compare them, we have to also compare the assays. Let's remove the duplicates here. In this case, we have duplicates only here. So it's the only one. So we can left only the first. Okay. So now let's find the duplicates so we will know which compounds we have both for human and mouse version of the enzyme. So here go to Home, Conditional Formatting and highlight duplicate values. Great. As you see, we have data for the same compounds, which are now indicated in red, both for human and mouse. Now we can apply filters to remove those compounds for which we have data only for human or for mouse, but not for both types of receptors. So for this, apply filters. And now filter by color, no fill. So in this way, here we can clear these fields. Now clear filter. And again, do the same for the second part. No fill. So in this way, we left only the compounds that are common for both types of receptors, human and mouse. Okay, so now remove the filter. And now we can sort the compounds by alphabet. Great, but still it's spreaded. Okay, so now we have both of them here. We have now only the compounds for which bioactivity data is present for both versions of the receptors here. So now we can remove one of them. The first one is for mouse. second one for human. As you can see, the first compound has the same affinity for both receptors, but all other compounds have manifold different activity. So for these compounds, we have a species-specific affinity. Now, if you proceed and carry out the exercises, you will learn how to prepare the spatial 3D structural models of complexes of receptors or enzymes with their ligands. So that way you can find the molecular basis, structural basis of species-specific affinity. Now, when we have the list of compounds, we may be interested in preparing the models of complex of these compounds with the corresponding proteins. The easiest way of finding the target protein structures, of course, is to use UniProt and Protein Data Bank. Let's start with UniProt. Put it here again, name retinoid acid X receptor gamma, and press enter. If you don't want to use the advanced search, of course. Okay, so here we have a lot of results, but let's leave only reviewed. Still we have a lot of them, 7. So here you have in the field of organism a list of organisms for which we have the sequences of retinoid acid X receptor gamma subunit. Here we have the Homo sapiens and Mus musculus, in which we are interested are. So let's open human and mouse versions. As you remember, in UniProt record page we have the section called structure. Now, when we moved here, we see that there is one 3D structure for a Homo sapiens type of the receptor with this code. So if you click on Protein Data Bank link, you directly move there and you see that it's a human retinoid acid receptor gamma ligand binding domain. So you can use it directly for docking studies, find out the spatial basis of species-specific ligand binding. And now let's move to mouse version of the protein. Click again on structure. And as you see here, there are no experimentally solved structures. To proceed with such kind of analysis, you have first to construct the homology model. How to do this? You will learn in the next week. In the week 4, when you will study the impact of genetic variation on the protein 3D structure. Species specificity is one of the examples of genetic variation, yes, because we have the same protein but with slightly different sequences. For some of the proteins, there are, of course, 3D structures solved not only for human but also for bovine version of the enzyme, maybe a rat version, mouse version, and sometimes even other organisms like guinea pig or monkey. Another useful thing about the species-specific action of drugs can be found in the large database of species-specific gene expression signatures. It's called PharmOmics, so you can just put the name PharmOmics in Google and find it. Let's go there. Here we have a beautiful website. So just click on the picture. So now we are in the PharmOmics graphical user interface. Here we have the list of drugs for which there are different kinds of gene expression data. A lot of different drugs. Then here in the second part we will have one of 3 species for which this database is available. To be more specific, it's Homo sapiens, Rattus norvegicus, and Mus musculus. So only for 3 organisms you can find data on drug-induced gene expression. In the third part, you can also select the kind of organ or organ system. This database allows you to find the not only species-specific data, but also tissue-specific data. And when you find it, you can retrieve more details, and also there will be a link to other database, to original data. Okay, let's find the data for one of the drugs. For example, we can find data for didanosine. Here we have data only for Rattus norvegicus in the liver. So it means that up to date, there is only one study about didanosine-induced gene expression changes in the liver of rat, and that's it. So let's select the other drug. For example, loratadine is a widely known antihistamine drug. Here we have again only data for Rattus norvegicus, but this time for 2 different tissues. So let's check the tissue-specific data. Okay, now we have this data here. So in this table we have the drug name, in this case loratadine, the name of original study, drug matrix, the tissue or organ. In our case we have 2 tissues, liver and cardiovascular system, only for the rat. And in this case, the link is not active, so it's not the best choice. Again, let's look for other one. So for isoniazid, it's one of the popular anti-tuberculosis drugs. We have bioactivity data for all 3 organisms, for human, mouse, and rat. And the data about gene expression changes after the injection or administration of these compounds only for liver. So we have 6 studies, all for isoniazid. So actually this data were accumulated just in 2 studies with these names. First study accumulated gene expression data only for the mouse, while the second one for human and for the rat. So in this case we have the links only to mouse gene expression data. So let's use it. It will lead us to Gene Expression Omnibus, one of the databases within the National Center of Biotechnological Information from the USA. This record page has again a lot of subsections, like title of the original study, then the organism, of course it's mouse, because we selected this, type of experiment, in this case it's expression profiling by microarray, short abstract, a brief description of the design, the authors and original publication, and here we have more specific data. So for this study there are 235 samples received, accumulated with one platform. So if you're interested in, you can proceed and look for the specific data. So this is the end of this short tutorial, and now you can proceed to further reading, case study, and also exercises. Good luck and see you later!